Hey there, and once again, welcome back to Mastering Medicine video lectures. In previous lecture, I talked about the structure and functions of the cell membrane. Today, I'm going to talk about an important function of the cell membrane, which is cell membrane transportation. During this lecture, firstly, I'm going to talk about different types of cell membrane transportation. For example, I'm going to talk about passive cell membrane transportation and its subtypes. For example, simple diffusion, osmosis, facilitated diffusion. And also, I'm going to talk about active cell membrane transportation and its subtypes. For example, symporters, antiporters, primary active cell membrane transportation, secondary active cell membrane transportations, and so on. And then I'm going to talk about bulk transportation and its subtypes, for example, exocytosis, endocytosis, and so on. And a little bit about the difference between pumps and channels. And also I'm going to talk about the structure and function of an important pump, which is called sodium potassium pump. Okay, its function and its structure, and also an important drug which affects sodium potassium pump, which are called as a group digitalis. Okay, so you don't need to write these notes to illustrate these illustrations. Go to description box and click on website link and download our today's video lecture free PDF lecture notes. Let's get started. So first of all, what is membrane transportation? As you know from previous lecture, this is a cell. Okay, a cell is surrounded by a cell membrane. So when molecules or substances move from the inside of the cell to the outside of the cell, or molecules or substances move from the extracellular area to intracellular area, this mechanism is called cell membrane transportation, and a cell needs for this mechanism, because destruction of this mechanism leads to destruction of cell, okay, or cell membrane. So first of all, cell membrane transportation is actually three types. One type is called passive cell membrane transportation, next type is called active cell membrane transportation, and third type is called bulk transportation. What are the differences between passive and active transportation? Actually, bulk transportation is under the category of active transportation. Okay, one important point between these two are why this is called passive and why this is called active, because in passive cell membrane transportation, when molecules move through the cell membrane, they don't use energy or ATP. There are a lot of types of energy, but which energy is here important? Biological energy or ATP. Okay, and in active transportation, and in active transportation, actually this transportation use ATP or biological energy. Okay, so here let me talk firstly about the passive cell membrane transportation. Actually, we have three types of passive cell membrane transportation: simple diffusion, facilitated diffusion, and osmosis. I'll talk about all of these one by one. Okay, first of all, what is simple diffusion? Simple diffusion means diffuses of substances, simply moving of substances simply. Okay, for example, we have some conditions for simple diffusion, that under these circumstances the molecules will move through the cell membrane by simple diffusion. For example, the first condition is that you know that in simple diffusion we don't use ATP or energy. This is one condition. Second conditions, in simple diffusion, substances move from high concentration gradient to low concentration gradient. What does it mean with concentration gradient? Concentration gradient means that the differences between amount of substances between two areas. Okay, for example, this is oxygen. These oxygen molecules are more outside of the cell than the inside of the cell. So here oxygen molecules move from high concentration gradient to low concentration gradient. It means from the outside of the cell to the inside of the cell. So another point is that in simple diffusion, molecules move from high concentration gradient to low concentration gradient. This is very important. Okay. Another important condition for simple diffusion is that molecules which move by simple diffusion, these should be lipid soluble. These should be lipid soluble. For example, oxygen is lipid soluble. If they don't be lipid soluble, they can't move by simple diffusion. So third condition for simple diffusion is that molecule should be lipid soluble. Okay. And another important condition for simple diffusion is its molecular weight. Okay, its molecular weight is so important because large substances or molecules can't cross the cell membrane by simple diffusion because cell membrane is semi-permeable. It don't allows anything to go inside or from inside to the outside. So it should be small. So it should be small molecular weight. Okay. And under these circumstances, under these conditions, molecules will move from high concentration gradient to low concentration gradient by simple diffusion. Yeah, there are some conditions that speed up the simple diffusion or slows the simple diffusion. For example, temperature. What? Temperature. For example, temperature degree, high temperature degree causes the increase of simple diffusion. Why? Because in temperature molecular movement increases, and due to increases of this molecular movement, molecules should pass through the cell membrane so fastly. So high temperature causes more simple diffusion. Okay. And another one which causes uh, more which causes simple diffusion to be more, this is molecular weight. Small substances, small things moves through the cell membrane by simple diffusion so fastly. Okay. So uh, less small molecules moves through simple diffusion more fastly. Okay. And also there is another important condition which is called membrane thickness. As you know, for example, when I thick this membrane, so molecules movement through the cell membrane got slowed. Okay. And this was a lot of about simple diffusion. Okay. As much concentration gradient goes up, simple diffusion got increased. Okay. And this was about simple diffusion. Let me talk now about facilitated diffusion. Okay, let me talk about facilitated diffusion. What does it mean? It means that something or someone comes here and it will facilitate or simplify our diffusion through the cell membrane. For example, what is the big difference between simple diffusion and facilitated diffusion? Both are parts of passive transportation, but in facilitated diffusion we use proteins. In simple diffusion we don't use ATP. Here also we don't use ATP, but in facilitated diffusion we use a proteins. For example, this is for example glucose. Okay, this for example glucose. When glucose moves from the extracellular to area to the intracellular area, it moves through these proteins, okay, through these transporters, through these facilitated proteins, and that's why we call this type of transportation facilitated transportation. We don't use ATP here, okay. This is one difference between facilitated diffusion and simple diffusion. Otherwise, it also here in facilitated diffusion we also move substances from high concentration gradient to low concentration gradient. And also here molecular weight is also important. Here molecular weight is also important. And also here we don't use ATP. In these things, simple diffusion and facilitated diffusions are same, okay. But what are the differences between these two? The first difference, that in facilitated diffusion we use proteins. Here we don't use proteins. The next difference, important difference, is there is the uh, thought of saturation, okay. What's saturation? For example, in simple diffusion, as amount of substances increase, so simple diffusion got fast, okay. But here in facilitated diffusion, as substances increase till some time, diffusion increase, but after some time, when concentration of molecules increase, movement of substances or diffusion of substances don't increase. Why? here here is a graph, okay. For example, here is concentration, and here is for example movement of substance or diffusion of substance. Here in simple diffusion, as concentration increase, movement of substance also increased, okay. But in facilitated diffusion, when concentrations increase, after till some time diffusion also increase, but after some times the graph will be flat. Why? Because for example, I have 100 channels of proteins here, okay. And in this same millisecond, I have 1000 molecules outside of the cell. So in this same milliseconds, 100 substance moves through the cell membrane, okay. These all 100 channels got saturated, but this 900 substances can't move through the cell membrane, because I don't have door to move these things from this area. This saturation concept is so important. For example, in diabetic mellitus patients, when we check urine of the patients, we will find glucose in urine, okay. But in normal person we don't have glucose in urine. Why? Because in normal person, when we filter glucose through the kidneys, so all glucose will be reabsorbed after some time by the kidneys, okay. But in diabetes mellitus, because filtration of glucose is so high, we don't have more pumps to reabsorb this glucose. So that's why we miss some glucose or some amount of glucose to urine, okay. So I don't go deep to this concept. That was about facilitated diffusion. There is one important thing, that sometimes facilitated diffusion is through carrier proteins, and sometimes facilitated diffusion is through channel proteins. What are differences between channel proteins and carrier proteins? This is so easy. First difference, channel proteins don't go to transformational chains, but carrier proteins go to transformational chains. For example, here is this pump. In step 1, this pump take a molecule. For example, this is gluta1 or glucose transporter type 1 for glucose. So first it will take a glucose, and step 2 it go to some transformational change, and in step 3 it will release this glucose inside of the cell. So this is carrier protein and this is channel proteins, because they go to transformational change and they don't go to transformational change. This one difference. The second important difference between channel and carriers are that channels in 1 millisecond transport around 1000 to million molecules or substances, but carriers, proteins, transport substances in 1 second around 100 to thousands molecules. Okay, so that was a lot about facilitated diffusion, different between simple diffusion and facilitated diffusion. Don't forget two differences. In facilitated diffusion we use proteins, in facilitated diffusion we have the concept of saturation. Okay, but in simple diffusion we don't use proteins and also we don't have concept of saturation.